Today we will start our discussion on the cestodes. We have seen in the previous lectures that the phylum Platyhelminthes that can be divided into two classes. One is the cestode class and another one is the trematodes. So there are morphological differences in between two classes of worms. The cestodes are also called the tapeworms because their body is flat and tape-like with multiple segments. The segments are united together to form a long tape-like worm. That's why it is called the tapeworm. The body of the cestode is tape-like and segmented, and the body is mainly divided into two parts. One is the head and another one is called the extension from the head, that is called a segment or proglottids. There can be multiple segments in a cestode body, and the proglottids, new proglottids arise from the head side. So the gravid and the ripe proglottids is at the distal end of the cestode's body. The head of the cestode has some special features that allows the cestode to be attached with the intestinal walls. The head of the cestode is equipped with hooks and suckers, and this is the distinction with the trematodes, which have suckers but they do not have any hooks. But the cestodes have both hooks and suckers with which it is attached to the intestinal wall. The head is also called the scolex. So to identify the cestodes, the special feature of the scolex is also important, with hooks and these suckers. The proglottids, the distal proglottids is the ripened one, and it contains multiple eggs. And at one point of the life cycle, the distal proglottids is detached from the whole body, and the eggs are liberated, and these eggs are infective form for the intermediate host. If we look through the image of a cestode, this is a graphical presentation of a cestode. You can see that the scolex, or the head part, and from the scolex there are a number of proglottids are formed, and the distal proglottids from the scolex, that means the last one is the gravid one. According to their maturity, the scolex are arranged, and the last one is the gravid with having many eggs. And you can see also the special features of the scolex having hooks and suckers. So the new proglottids, they grow from the scolex, and so the distal proglottids is the oldest and the gravid one, and this distal proglottids contain the mature eggs. So in case of some cestode, the whole proglottids may be found in the stool of the definitive host, or sometimes the egg can be found in the stool of the definitive host. Now let us see some important examples of cestodes which are medically important, and we will discuss in detail their life cycle and laboratory diagnosis. So medically important cestodes are: number one, the Tinea saginata, it is also called the beef tapeworm. Then Tinea solium, this is called the pork tapeworm. Then Echinococcus granulosus, this is the causative agent of hydatid cyst, and this is also called dog tapeworm. And there is Echinococcus multilocularis, and also the Diphyllobothrium latum, it is called the fish tapeworm. So the tapeworm can be named depending on their scientific name, and also in generally they are sometimes called on their intermediate hosts. So for the cestodes, the intermediate host can be different, like cattle, pig, fish, or dog. They harbor the larval stage of the parasite, and the definitive host is the human, where the adult worm lives in the intestine of the human being, and humans are infected by ingestion of the larval stage from the intermediate host. So human can be infected by two ways. One is by ingestion of undercooked meat of the intermediate host, which contain the larval stage of the parasite, and also by accidental ingestion of the eggs directly contaminated with the human feces. And eggs causes infection in case of hydatid cyst and the cysticercosis. Now let us compare the some important characteristics between the Tinea saginata and Tinea solium. These two tapeworm causes most of the infection in human. The Tinea saginata is also known as the beef tapeworm because the intermediate host is the cattle, and the Tinea solium is called the pork tapeworm. The larval stage of the Tinea saginata is called Cysticercus bovis, and in the Tinea solium it is called the Cysticercus cellulosi. The length of the Tinea saginata is much more. It is about 10 to 15 meter long. It may be more than that. And in case of Tinea solium, it is a bit shorter than the Tinea saginata. It is less than 7 meter long. Then if we consider the proglottids of Tinea saginata, it has more than 15 uterine branch in its gravid proglottids. In case of Tinea solium, it is less than 13 the uterine branches, and this is an identifying point. If we examine the proglottids under microscope, we will see there will be more than 15 uterine branch in case of Tinea saginata, and in case of Tinea solium it is less than 13. There may be 1000 to 2000 segments of proglottids in Tinea saginata, but in case of Tinea solium it may be less than 1000. So these are the some important distinguishing feature between the Tinea saginata and Tinea solium. Also there is difference in the scolex also. The scolex or head in case of Tinea saginata do not have hooks or rostella. They have only suckers. In case of Tinea solium, it is equipped with both the rostellum or hooks and also the suckers. And there may be number of differences in this between two, but these are the important one. There are some other important features of these two tapeworms to distinguish or detection of them. The proglottids of the Tinea solium usually expelled in a group, not singly. The gravid proglottids detach from the adult worm in a group. There are multiple segments detached at a time. But in case of Tinea saginata, usually only one gravid proglottids detach from the adult worm. And this single proglottids that detach from the adult worm is usually expelled actively from the anus. The carrier or the infected person even can notice that the proglottids of the Tinea saginata is coming out through the anus so actively and forcefully the person can even notice. But in case of Tinea solium, this is usually expelled passively, and usually they are in a group. And the cystic stage of Tinea saginata is called cystosarcosis, Cystosarcus bovis, and in case of Tinea solium it is called Cystosarcus cellulosae. This is the larval stage. The larval stage forms a cystic stage. The cyst means it is a water-filled sac-like structure within which the scolex is invaginated.